Okay. So the antibodies are produced by the B cells. The activated B cells will which first change or differentiate to plasma cells. And then we use the word plasma cells secrete antibodies, right? Acha. Now, when we were doing biological molecules, we studied that there are two forms of proteins, globular proteins and fibrous proteins. In the examples of globular protein, I don't know whether you guys remember or not, but the most common or most important globular protein that we did was hemoglobin. And then to just state as examples, we said enzymes, antibodies, all these are also globular. And then in fibrous proteins, we discussed collagen and keratin and all. So antibodies are also globular proteins. Now, when I say globular, what do you mean by that, Amna? What
It's
is
the
the word?
proteins.
Yes, yes, yes,
It's the
Pita?
proteins that are in spherical form.
In spherical form. Okay. Uh, Besides spherical form, what else comes to your mind? The proteins which are water soluble or water insoluble?
Miss water soluble.
Water soluble. Very good. And what would you imagine that these proteins have got their hydrophilic R groups pointing to the center or to the outside?
It's outside.
Outside, right? Of course, outside, because it's the outside. Yes, you can mute yourself. It's the outside part of these proteins which is facing the aqueous environment of the cell. So hence it needs to be made up of hydrophilic or water soluble amino acids so that the protein can function and take part in various chemical reactions. Okay. So antibodies are globular glycoproteins. Now there's something very special. They have a glyco component also attached to it, which means that they have a polysaccharide portion with a quaternary structure. Okay, Parishe, what do you mean by the word quaternary? How will you define it?
Is a structure that is formed by more than one polypeptide chain.
Very good. Any protein that is made up of more than one polypeptide chains is classified as quaternary. So, since hemoglobin is made up of 4 polypeptide chains, so it is also quaternary. Myoglobin is also quaternary. And antibodies are also quaternary, right? How many chains? They are made up of 2 heavy chains and 2 light chains. Okay, I'll show you the diagram and then you will soon realize what it is. And those chains are made by disulfide. They are held together by disulfide bonds. Now, instead of me just giving you details in bullet points, it is better that I show you the diagram. Okay. So there you go. 4 polypeptide chains, and I'm going to use my cursor to show you those chains. Chain number 1, chain number 2, chain number 3, and chain number 4, right? Each chain is made up of a variable region. You can see this is the variable region. So for example, if I take into account this chain, ठीक है, तो इस chain का एक variable region है और एक constant region है, right? Similarly, this chain also has a variable region and a constant region. So basically, if I say what are the variable regions, let me just put a circle on this. These are the variable regions. This is that region which makes an antibody unique. Okay, this is what makes an antibody unique. So this means when I say that I have in our body, as I was explaining you last time, that when the B cells are maturing in the bone marrow, millions and millions of varieties of B cells are made, each capable of producing an antibody against an antigen to which it has never been exposed. अभी वो expose ही नहीं हुआ B cell, लेकिन उसके पास ये capability है कि it can respond to an antigen and make the specific antibody. तो कैसे हो सकता है possible? All because of the variable region. ये different है. इसका मतलब ये जो region है वो हर antibody का different है because it will bind to the specific antigen. ठीक है? अच्छा, light chain और heavy chain देख लेते हैं. अच्छा, now this is one light chain. ये पूरी जो है वो light chain है. And this is also another light chain. ठीक? This is one heavy chain. Of course, heavy chain is heavy means it's bigger in mass. So this is one heavy chain. And this is another heavy chain. ठीक? So two heavy chains and two light chains, which makes up a total of 4 polypeptide chain. और जब हम कहते हैं polypeptide, तो इसका मतलब है कि इसमें बहुत सारे amino acids होंगे ऐसे करके. Lots of amino acids. ठीक है? Over here also lots of amino acids in its primary sequence. लेकिन जो यहाँ पे amino acids हैं, they are different. They are not same. They are different in different kinds of antibodies. ठीक है? अच्छा, now to hold two heavy chains together and to hold a heavy chain and a light chain together, we have these bonds, जो कि disulfide bonds हैं. And you know from your prior knowledge when you were doing biological molecules, they are the very strongest bonds that happen to occur in protein molecules, and they're very, very difficult to break. ठीक है? अच्छा, so they help in sort of fixing these polypeptide chains together, and these are the disulfide bonds which make the antibody molecule to be very, very flexible. क्योंकि antibody का तो काम ही यही है ना? Because what we did last time was this. At some places, the antibody wants to bind to a toxin. At some places, it wants to stick to flagella. At some places, it wants to clump the bacteria. At some places, the antibody wants to punch holes in the cell wall of bacteria. कहीं पे कुछ कर रहा है, कहीं पे कुछ. तो इसको ये इतनी सारी एक्टिविटी करने के लिए it needs it to be very, very flexible, ताकि इधर उधर आराम से अपना काम कर सके, right? So this flexibility is given by the disulfide bond. It holds these chains together and it makes the antibody flexible. Acha. As it says, the hinge region gives flexibility in binding to antigen. तो यही hinge region है, ठीक है? Then antibodies are also covered by a chain of sugar molecules. Sugar is always sticky. अगर हमारे हाथ पे भी चीनी लग जाए तो कितना sticky feel होता है, right? Similarly, the attachment of sugar molecules onto the antibody makes it sticky, and we want antibodies to be sticky so that they can attach to the bacteria, viruses, toxin, etc., etc. Which is why हम इस antibody को क्या कहते हैं? हम कहते हैं it's a glyco. So glyco is this part, and it's a protein. Protein ये सारा part है. ठीक है? So, any questions up till now as far as the structure of the antibody is concerned?
No, miss.
No? Acha, let me just show you a question from a past paper. ये आपका एक question है. It says, antibody against measles are produced by plasma cells during an immune response, right? Figure 2.1 shows a diagram of an antibody molecule. Part A, part C, and part B is labeled. Okay, so what do you think you should name part A as? Yes, Amna? Which region is part A of this antibody molecule? Parisha?
Is the light polypeptide chain?
No. Light polypeptide chain, तो light तो ये है, लेकिन इसमें heavy भी है. लेकिन हमने इसको क्यों circle किया हुआ है? What is this? Is this the constant region of the antibody or the variable region?
The variable region.
Variable region, ठीक है? A आपका variable region हो गया. B कौन सा region है? What is B?
It's a bond.
The bond, or you can say it's the hinge region. Let's go back and see the antibody. Wait a minute. This is the variable region. ठीक? This is the hinge region that holds the polypeptide chains together, and this is the constant region. ये जो आपका ये जो पूरा stem है ना, that's the constant region. ठीक है, अब दोबारा से उसी डायग्राम पे चलते हैं. A, B and C. A is the variable region, B is the hinge region, and C is the constant region. And then the examiner wants you to explain the functions of part A, B, and C. So instead of asking you questions, I am sort of sharing the questions with you, the answers with you. तो आपको पता चले कि A, B, and C का क्या function है, ठीक है? This is region A, ये वाला, जो कि, sorry, which is the variable region. This is region B, which is the hinge region. I don't know why they are saying ignore reference to hinge, but basically they just want you to say it's disulfide bond. And this is region C, which is a constant region. Now I want all of you to read these three descriptions very, very carefully, and then I will discuss with you. Girls, do let me know if you're done.
Miss, I'm done.
Okay, what about you,
I'm
Parishe?
done.
Acha, okay.
I'm done as well.
Okay, fine. Right. So we have this constant region. First of all, sorry, the variable region. So it says कि आप देखो, आप इसकी wordings देखो. It says कि आपको कैसे इसको describe करना है. It's the region that binds or attaches. Use the word binds or attaches to the antigen. जाहिर है, ये ऐसे है, ठीक है? ये आपकी antibody ऐसे है, for example. तो ये जो region है, ये वाला, ऊपर वाला जो V shape है, इसका shape भी V है, and we call it variable region. So this is the variable region. Why? Because this is going to bind to a specific antigen जो उसी shape का हो. Let's say कि antigen का ये shape है, ये उससे bind करेगा, ठीक है? Don't use the word receptor, don't use the word fit and whatever, a complementary, active site, इस तरह के words नहीं use करने हैं. सिर्फ आपको ये कहना है कि this region is very specific because it binds or attaches to the specific antigen. ठीक है? अच्छा, अब आते हैं उस पे. The hinge region or the disulfide bond, its purpose is that it holds the polypeptide chains together, the heavy chains and the long chains together. ठीक है? and it maintains the tertiary or the quaternary shape of the what do you say, the antibody. क्योंकि when we were doing tertiary structure ना, हमने कहा कि primary structure तो sequence of amino acids है. Secondary structure is only because of the regular pattern or arrangement of the hydrogen bonds. जो आपका tertiary structure होता है वो ionic bonds, disulfide bonds की वजह से होता है. So when you are describing the disulfide bond or the hinge region, आपको उसके दो function याद रखने हैं. कौन से? It's good that this class is being recorded और आप लोग अपने notes में भी लिख सकते हो कि the function of the disulfide bond is two functions. ठीक है, कौन सा function है? Function number 1, it holds the polypeptide chains, heavy chains and the long chains together, heavy chains or the long chains together, and it maintains the tertiary or quaternary structure. दोबारा से देखते हैं क्या ये वाकई ऐसे कर रहा है. ये देखो, अगर आप hinge region को देखो तो ये जो आपका hinge region है ना वो ये वाला है. This is the hinge region. तो अगर आप इसको गौर से देखो तो ये जो hinge region है, it is holding these two heavy chains together. Not only this, but it is also responsible for the tertiary or the globular shape of the antibody. ठीक है, फिर हम आते हैं last वाले part पे, which is the constant region. Constant region कौन सा है? Let's connect this with one of the diagrams that we had done when we were doing phagocytosis. जब हम phagocytosis कर रहे थे, so there was a diagram जिसमें कि ऐसे करके आपका phagocyte बना हुआ था. I hope you girls remember. ठीक है, ये phagocyte membrane बनी हुई थी, right? There was a bacterium जो कि directly आके यहाँ लग रहा था, and there was another bacterium which was tagged by an antibody. ठीक है, ऐसे करके antibodies को tag करके लेके आ रही थी, and here there was a receptor. ये यहाँ पे आएगी antibody, and it will sort of tag this bacterium and bring it close to the phagocyte so that it can be easily phagocytized. So the function of the constant region is कौन सा वाला? ये वाला region. ये हम Y shape बनाते हैं ना, तो ये जो है ये variable है, ये वाला जो डंडा है ना Y का, that's the constant region. So the constant region binds to the receptors on the cell surface membrane of the phagocytes or the macrophages, and it will help in marking or tagging the antigen. जो भी antigen है, चाहे वो bacteria है या virus है, marking or tagging the antigen so that it can be easily phagocytized. ठीक है? Let me show you this thing once again in the diagram that we have done before. We have done this over here. इसको आप गौर से देखो. ये मैंने आपको बताया था कि either the bacterium can directly attach to the phagocyte cell surface membrane through its receptor, or there is a receptor for the antibody marker. ये आपको नजर आ रहा है, ये antibody खींच के ला रही है, mark करके ला रही है किसी bacterium को, and it is going to bring this, sort of tag this bacterium and bring it close to the phagocyte so that it can be easily phagocytosed. G girls, is this concept understood?
Yes, Miss. Miss, I had a question that I forgot to ask earlier.
Yeah, sure.
Miss, is there any blood supply or a capillary or anything going into the bones?
Is there any blood supply? Repeat your question.
Miss, is there any capillary or vessel going into the bones?
Into the bones? Yes, of course. But there's no blood supply in the cartilage.
Miss, but I think their bones are like, they didn't have any blood supply or anything.
Acha, if a person breaks his bone, doesn't he start bleeding? He can bleed also, isn't it? Of course.
Okay, Miss.
If a bone doesn't have a blood supply, then how do you think the bone is going to grow? And in the case when a person has his bone fractured, तो उसकी healing ही blood supply से होगी. Remember one thing, that no tissue of your body can heal without the blood supply. और हमारे body में दो ऐसे tissues हैं जहाँ पे कोई blood supply, मतलब capillaries नहीं है. One is the cornea and the other is the cartilage. अच्छा, तो फिर सवाल पैदा होता है, हाउ डज कॉर्निया गेट इट्स न्यूट्रिएंट्स? सो कॉर्निया गेट्स न्यूट्रिएंट्स थ्रू द एक्वस ह्यूमर। सिमिलरली जो आपका कार्टिलेज है उसको न्यूट्रिएंट्स मिलते हैं साइनोवियल फ्लूइड से। इसी वजह से द डॉक्टर्स से दैट व्हेन योर जॉइंट, कार्टिलेज हमेशा जॉइंट के पास होता है, अगर आपको मसल की इंजरी हुई है तो वो जल्दी हील हो जाएगी, लेकिन इफ योर जॉइंट इज इंजर्ड, कभी कभी प्लेयर्स के, क्रिकेटर्स के, फुटबॉलर्स के जॉइंट में इंजरी हो जाती है, सो फॉर दैट देन दे बिकम अनफिट फॉर अ वेरी लॉन्ग टाइम। तो जॉइंट टेक्स अ वेरी लॉन्ग टाइम टू हील क्योंकि उसमें कैपिलरीज नहीं होती। ठीक है, ऑफ कोर्स बोन्स हैव ब्लड सप्लाई। अच्छा, so we have done the structure of the antibody, and now let's move on to, now this means we are done with B cells, and today now we'll move on to T cells. ठीक है, T cells की तरफ चलते हैं। Okay, in fact I think I want you, ये slides जो है ना, they are made from the old textbook, so I want you to actually read the new textbook because it has been explained really very well. तो हम आते हैं T cells पे। we're done with B cells, just to show you कि हमने क्या क्या किया है। we did all this, ठीक है, we started with lymphocytes, we did B lymphocytes, ये सब हमने कर लिया है। we did selection and expansion, plasma cell, memory cell, ये भी सब कर लिया हमने। primary immune response, secondary immune response, and now हाँ, B cells and antibodies भी कर लिया। variable region हमने अभी अभी किया। if you want you can go through the definition of the variable region. Your textbook is saying it is a region of an antibody molecule composed of parts of light chains and heavy chains, ठीक है, that form the antigen binding site. The amino acid sequences inside the variable region, they give specificity to the antibody. अगर आपसे एग्जाम में कोई पूछता है कि what gives the antibody its specificity and what makes an antibody very very specific, ठीक है ना, जैसे हम एंजाइम्स के लिए कहते हैं कि एंजाइम्स की स्पेसिफिसिटी उसका एक्टिव साइट है, सही। Similarly, the specificity of the antibody is its variable region, and what makes it specific? The specific sequences of the amino acids in the variable region. तो ये बहुत important है, these definitions, help you in building up your concepts, especially for paper 1. अच्छा, अब हम आते हैं, we did this also, right? We did this also, and now we come to T cell activation. I want you कि आप इस पैराग्राफ को बहुत अच्छे से पढ़ो। I'm giving you 5 minutes, ठीक? And then I will explain it to you. जो आपको नहीं समझ आया होगा, I'll try to make connections for you, right? So start, please. Start reading it. In fact, girls, it is Maghrib time over here, so I will take a break for my prayer. So this is अच्छा है कि आप पहले से पढ़ें, because ये होता है कि it only one person is talking and it becomes boring for the student to grasp these concepts. So it is page number 275 and page number 276. See if this can make sense to you. And after 10 minutes, I'll continue the class. And if I want to extend the class today till 7:10 p.m., is it okay for both of you?
Yes, miss.
Okay.
Yes, miss.
So I'm taking a break of 10 minutes, and please continue reading. Yes, Amna and Parisha, can you hear me?
Yes,
Yes,
miss.
Okay. So, are you done reading with these two?
Yes, miss.
Acha, okay. So, first let's start with this. So, we are done with the B cells and now we move on to see how the T cells are activated, what actually happens, as far as their clonal selection, how many kinds of T cells are there. So, it's a bit different from B cells, right? Acha. So, the discussion begins with the fact that once the T cells are matured, इसका मतलब हुआ कि the T cells we've already known, we've already studied this before, that the T cells mature in the thymus. Okay? So once they are mature, they have specific cell surface receptors known as T cell receptors. And these T cell receptors have a shape very similar to that of the antibodies. And they are specific to one antigen. T cells are activated when they recognize this antigen on another cell of the host that is on the person's own cells. Mostly, if you remember, I think I've told you this before, that such a cell, which is a person's own body cell, which can actually present the antigens by first cutting them off partially and then finally presenting it on its surface, is a macrophage. Macrophage ये कर सकता है. क्या करता है? सबसे पहले, it engulfs a pathogen, cuts it up to expose the pathogen's surface molecules, or it may be a body cell that has been invaded by a pathogen and is similarly displaying the antigen on its cell surface membrane as a kind of help signal. The display of antigens on the surface of the cells in this way, this process is known as antigen presentation. I'll draw a diagram to show you how it is. The T cells that have receptors complementary to the antigen respond by dividing by mitosis to increase the number of T cells. T cells go through the same stages of clonal selection and clonal expansion as B cells. Okay, so let me show you this diagram. I have it, I have made it. Yes, it is here. Just a minute, guys. Okay, so I think I'll have to draw it. So your T cells, this is your thymus gland. Okay, and your T cells have matured inside they contain mature T cells, the thymus. And let me draw one single mature T cell. Let's say it's this one single. Right? Now, this mature T cell has got receptors on its cell surface membrane, which are known as the T cell receptors. The shape of these receptors is similar to the antibody. Not the antigen, but the antibody. Don't forget, girls, that the T cells don't make antibodies. It is the job of the B cells, which differentiate into plasma cells, and the plasma cells secrete antibodies. T cells nahi banate antibody. Lekin, एक interesting बात यह है कि the T cells receptors shape is similar to the shape of the antibody. ठीक है? Through these receptors, the T cell will recognize a particular antigen. ठीक है? I'm going to write down these points here. Through these receptors, the T cells will recognize the antigen. Antigen recognition will take place. Achha, how is the T cell going to recognize the antigen? The antigen recognition takes place through the antigen presenting cells. And in short form, you can call them APCs also. APCs. What are the two possible APCs, at least for your level, for AS level, और भी बहुत सारे हैं, but they can be the macrophages, or they can be the body's own cells. Macrophages, कौन से वाले macrophages which has engulfed a pathogen, cut it, and displayed the antigen on its surface. In fact, I think I should show it to you in the form of a diagram. ठीक है, ये आपका macrophage हो गया. It has a kidney-shaped nucleus. So the macrophage has engulfed some antigen and then उसको cut करके उस antigen को इसने display कर लिया है अपने surface पे. ठीक? Or it can be any random cell of your body, कोई सा भी cell हो सकता है, which has been invaded by a pathogen. Let's say कोई pathogen है अंदर, and then it is also showing the pathogen or the antigen on its surface. So what does the T cell do? The T cell will recognize, the T cell receptors will recognize this antigen. Okay? Once the recognition takes place, what happens? There is a cell signaling mechanism that is activated. कैसे? ये देखो ये क्या लिखा हुआ है यहाँ पे कि once this takes place, the antigen which is being displayed on the surface of the macrophage, it is considered to be as a kind of a help signal, right? The display of the antigens on the surface of the cells in this way is known as the antigen presentation. The T cells that have receptors complementary to the antigen respond by dividing by mitosis to increase the number of the T cells. So what will happen now? This will be considered as a help signal. And this help signal will stimulate the mitosis of the T cells. T cell mitosis is stimulated. Lots of T cells made. So this means अब जब lots of T cell बन गए तो क्या हो रहा है असल में? Clonal expansion is taking place. We can say this was the clonal selection कि वो खास T cell recognize हुआ था. कौन सा खास? जो कि इस receptor से complementary हो. So this clone of T cell was selected and now this clone has expanded. Any questions? Yes, girls. I hope both of you can hear me.
Yes, miss.
Acha, okay. There's no problem in understanding? Sure. Now coming to the two varieties of T cells that we have. Now in our body, as opposed to B cell, जो कि एक ही variety का था, we have two kinds of T cells. One are the T helper cells and the other are the T killer cells. ठीक है, दो हमारे पास T cells हैं. What does the T helper cell do and what does the T killer cell do? The T helper cells are the kind of the T lymphocyte that secrete chemicals. ये chemical होते हैं जिसका नाम है cytokines, whose job is to coordinate the activity during the immune response. अभी हम इसकी डिटेल करेंगे कि कैसे cytokines जो है वो coordinate करते हैं activity during an immune response. ठीक है? और फिर T killer cell क्या करेंगे? They are another unique variety of T lymphocyte that attaches to cells directly, releasing toxic substances to kill वायरस इन्फेक्टेड सेल्स एंड कैंसर सेल्स। तो बेसिकली आपके जो टी किलर सेल्स हैं वो डायरेक्टली अटैक करते हैं किसके ऊपर? वायरस इन्फेक्टेड सेल्स पर एंड कैंसर सेल्स पे। सो हेंस द नेम टी किलर सेल्स। लेट्स सी व्हाट द टी हेल्पर सेल्स डू। I have slides to show you what the T helper cells are doing. Helper T cells, as your textbook is saying, they release very important chemicals known as cytokines, and cytokines के 3-4 important function है. Now you have to listen to this part very, very carefully. Cytokines are chemical substances which are released by the helper T cells. They are hormone-like substances. They can stimulate the B cells to divide, यानी कि B cell जो अभी हम पीछे कर चुके हैं, cytokines जो कि helper T cells रिलीज कर रहे हैं वो B cells को डिवाइड करने में हेल्प कर रहे हैं, डिफरेंशिएट करने में हेल्प कर रहे हैं इनटू प्लाज्मा सेल्स. ठीक? Not only this, the cytokines also help the B cells secrete antibodies. तो मतलब क्या हुआ कि cytokines are helping the humoral immunity to take place, because the immunity given by B cells is the humoral immunity. What do? B cells को डिवाइड करने में हेल्प करते हैं, डिफरेंशिएट करने में हेल्प करते हैं, सीक्रीट करने में हेल्प करते हैं. ठीक है? सो वेरी वेरी pivotal function of the cytokines. Let's see any more function. और क्या फंक्शन है? और फंक्शन ये रहा. The helper T cells, they secrete cytokines. So वो cytokines क्या करेंगे? They help the macrophages to increase the phagocytosis. बड़ी अजीब सी बात है कि मैक्रोफेजेस तो अपना काम कर रहे हैं, फैगोसाइटोसिस कर रहे हैं, लेकिन उनको फैगोसाइटोसिस में हेल्प करते हैं साइटोकाइंस. एंड हु इज रिलीजिंग साइटोकाइंस? हेल्पर टी सेल्स आर रिलीजिंग साइटोकाइंस. ठीक है? एंड द साइटोकाइंस आल्सो हेल्प द किलर सेल्स टू डिवाइड बाय माइटोसिस एंड डिफरेंशिएट. तो इसका ये मतलब हुआ कि हेल्पर टी सेल्स आर वेरी वेरी इंपॉर्टेंट. व्हाई? बिकॉज द हेल्पर टी सेल्स are helping what kind of cells? The helper T cells are helping B cells. The helper T cells are helping macrophages, and the helper T cells are helping killer cells. तो इसका मतलब हुआ helper T cells are playing a role in humoral immunity, which is a kind of specific immunity. हेल्पर टी सेल्स आर हेल्पिंग मैक्रोफेजेस, तो फिर ये नॉन स्पेसिफिक इम्यूनिटी में भी इनका रोल है. और वो किलर टी सेल्स को हेल्प कर रहे हैं. तो किलर टी सेल जो इम्यूनिटी देते हैं ना, उसका नाम है सेल मीडिएटेड इम्यूनिटी. और सेल मीडिएटेड इम्यूनिटी भी एक काइंड ऑफ स्पेसिफिक इम्यूनिटी है. एनी क्वेश्चंस गर्ल्स? ओके. अच्छा, अब इस डायग्राम को गौर से देखो. We said that the T cells, अब पीछे चलते हैं कहानी में, कि the origin of T और B cells is in the bone marrow. वो वहां पे immature होते हैं और वो डिवाइड कर रहे हैं माइटोसिस पे. When the maturation takes place, it is taking place in the thymus. Maturation का मतलब they develop receptors, और थोड़ी ही देर में इसके surface पे receptors आ जाएंगे, और इन receptors का shape जो है it is very similar to the antibody. ठीक? अच्छा, ये mature हो गया T cell. From let's say कि वो millions में है, लेकिन हम यहाँ पे 3 दिखा रहे हैं. So why is this mature T cell selected? Because this specific antigen has entered your body. Select हो गया ये. Selection के बाद some of them can, अच्छा selection से पहले भी ऐसे हो सकता है, depend करता है कि the mature T cells they circulate. एक दफा जब वो mature हो जाते हैं वो हमारे blood में circulate करते हैं. Some circulate as helper T cells, some circulate as killer T cells. डिपेंड करता है कि अब कौन सा वाला सेल जो है वो एक्टिवेट हो रहा है. हेल्पर भी एक्टिवेट हो सकता है और किलर भी एक्टिवेट हो सकता है. तो उसी तरह की जो है वो क्लोनल एक्सपेंशन एंड क्लोनल सिलेक्शन होगी. दिस डायग्राम इज टेलिंग यू दोनों चीजें आपको ये डायग्राम बता रहा है कि हेल्पर टी सेल भी एक्टिवेट हो सकता है, किलर टी सेल भी एक्टिवेट हो सकता है. In order to activate the helper T cell or the killer T cell, what do we need? We need an APC. And this is an APC. This is a cell, any random cell of your body, or a macrophage which is displaying the antigen on its surface. Depending कि इस antigen का shape क्या है, तो या तो ये किसी helper T cell को stimulate कर देगा, या फिर किसी killer T cell को stimulate करेगा. जो भी हो, right? Whatever the cell is stimulated, that specific helper or killer T cell will divide by mitosis. हो गया divide? अच्छा, इसके बाद बिल्कुल वैसे ही होगा जैसे B cells में हो रहा था. Some of the cells जो कि divide हो रहे हैं mitosis में, वो बन जाएंगे आपके memory cells. Those which are not forming memory cells, जो आपको foreign immunity दे रहे हैं, वो क्या करेंगे? They will secrete cytokines, जो कि helper T cell का काम है कि वो cytokines बनाएं. And I've already told you that cytokines सब कुछ कर सकते हैं. They can stimulate the B cells, they can stimulate the plasma cells, they can stimulate the macrophages, they can stimulate the killer T cells. सब कुछ कर सकते हैं helper T cells. Why? Because they are making a very, very precious substance which is known as cytokine. अच्छा, let's come to the killer T cell. Killer के नाम से पता चल रहा है कि directly kill करेगा. What kind of cells do these helper killer T cells kill? They can kill two kinds of T cells. ये बात हमेशा याद रखनी है. Number one, a virus infected cell. Number two, a cancer cell. Depends. ठीक है, देखते हैं ये क्या कर रहा है. Killer T cell ने अपना specific antigen recognize किया. Again, just like helper T cells, some of them will divide to become memory cells, and some of them will actually do the destruction of an infected body cell, a transplanted cell, or a cancer cell. How does it attack it? जो आपका killer T cell है, ये रहा killer T cell, तो उसके अंदर होते हैं vacuoles, ठीक है? A vacuole is shown, and those vacuoles contain deadly toxins. Those toxins, what do? They punch holes in the cell surface membrane of the infected body cell. Let's say कि ये आपका infected body cell है और इसको destroy करना है, तो killer T cell क्या करेगा? ये killer T cells के इस part को zoom करके आपको दिखाया गया है कि killer T cell के अंदर vacuoles हैं. Vacuoles are rich in toxins, and the toxin can directly punch holes in the cell surface membrane of the infected body cells. And so the killer T cell will directly destroy the virus infected cell, the cancer cell, or the transplanted cell. लेकिन इस killer T cell को भी ये cytokines help करती हैं, right? In fact, I'll show you a concept map over here. अगर हम overview देखना चाहें ना हर तरह की immunity का, तो हम ये कह सकते हैं कि whatever the immune cell is, every immune cell, whether it's a B cell or a T cell, starts from a precursor cell or an immature stem cell in the bone marrow. ठीक है, undifferentiated stem cells हैं. ठीक है, आपकी bone marrow के अंदर undifferentiated stem cells हैं. यही हमने माइटोसिस के चैप्टर में किया था. ठीक, ये undifferentiated stem cells differentiate हो रहे हैं, तो अब बन गया एक immature lymphocyte. अभी भी ये काफी undifferentiated है. Again, now some of them become B cells, some of them become T cells. तो जो B cells बनेंगे, they are responsible for the humoral mediated immune response. इसको हम कहते हैं HMIR. और जो T cells बनेंगे, they are responsible for the cell mediated immune response. ठीक, लेट्स सी क्या हो रहा है आगे. बी सेल्स तो आपको पता है, बी सेल्स को जैसे ही एंटीजन एनकाउंटर होगा तो बी सेल क्या कर रहे हैं? दे कैन मेक मेमोरी सेल्स, दे कैन लिव फॉर मंथ्स और इयर्स, और दे कैन मेक प्लाज्मा सेल्स दैट कैन सीक्रीट एंटीबॉडीज. टी सेल्स को देखते हैं क्या करते हैं. टी सेल्स कैन आइदर डिफरेंशिएट इनटू हेल्पर टी सेल्स और इनटू किलर टी सेल्स. अच्छा, suppressor T cells भी होते हैं, लेकिन they are not a part of your syllabus, so I'm not going to teach you about suppressor T cells. ठीक है, helper और killer. तो आपको नजर आ रहा है कि हेल्पर किसको हेल्प कर रहे हैं? बी सेल्स को भी हेल्प कर रहे हैं, हेल्पर टी सेल्स को भी हेल्प कर रहे हैं, और हेल्पर जो है वो आपका फैगोसाइटोसिस ऑफ phagocytosis by macrophages को भी हेल्प कर रहे हैं. Why? Because the helper T cells are making cytokines. Okay, girls. This is a question for you, and you are supposed to solve this and let me know the answers. And I'm waiting. Describe the modes of action of T lymphocytes during an immune response. Start, please. Girls, there is so much silence. It seems as if both of you are not in the class.
Yes, will we mention that the T cells can directly kill the antigen?
Yes, you can mention. T cells can directly kill them. So this, but in that case, then you will have to talk about which one, killer or helper?
Killer?
Okay, so I'm writing down your points and then I will compare it with the mark scheme. So killer T cells
Miss,
directly kill the antigen. Yes, Amna, you want to say something?
Miss,
Ji.
Miss, can we include cytokines?
Of course, you can include cytokines, but then you will have to talk about helper T cells, release cytokines. Yes. And what do cytokines do? So see, let's look at the question. If you look at the question, it is not specifying helper or killer cells. It's a general question, and it is generally asking you the modes of action of T lymphocytes during an immune response. So this means that the examiner has given you a free choice to talk about both kinds of lymphocytes. So yes, the answer is correct. But then of course, हर possible point यहाँ पे लिख दिया गया है ताकि आपको पता चले. So if a student wants to talk about helper T cells, then he will talk about secretion of cytokines. We know that those cytokines can activate B lymphocytes to make plasma cells. They can also activate the phagocytosis by macrophages. ठीक है? और अगर वो cytotoxic T cell भी इसका नाम है और killer T cell भी नाम है, दोनों नाम है. It can directly kill the infected cells, as Parisha said. It can kill tumor cells, and it can also kill the antigens, right? It can punch holes. Those substances which can pump the mechanism of killing by punching holes is through these substances known as perforins, right? Now there is another question. Wait a minute. I would like you to solve this question, please. During an immune, Ji beta.
Miss, can we talk about how T and B cells are divided, undergo mitosis to produce plasma cells and
Yes.
memory cells?
Yes. So, acha, let me go back to the question. Describe how mitosis is involved in an immune response. So we know that without mitosis, neither the B cells can get activated, nor the T cells can get activated. So this means both the B and the T cells require that mitosis should happen, right? It happens before maturation also, and it happens after maturation also. So look what's written over here. It's also a question of 3 marks. Mitosis of B and T cells occurs both in the primary and the secondary immune response, right? Why? Because selected B and T cells, which are specific to the antigen, they are selected and they divide by mitosis. Once they are selected, then what happens? The clonal expansion can take place. And yes, that's it. Idea that different kinds of immune cells can result, reference mitosis in memory cells for rapid secondary response. Acha. So what they're trying to say over here is that mitosis is an essential process that happens in both primary and secondary immune response. Okay? Without mitosis, the T and B cells cannot produce their response, and a certain variety of B or T cell needs to be selected. And then when we want, after selection, when we want to expand that particular clone of cells, then it happens through mitosis. I think it's enough for today, and I'm ending the class right now. We will continue on Wednesday, inshallah.